topic, that is cell and biological membranes, which you all have been studying through the previous years also. So it's just like a revision of the previous topic. Okay. Now, as you all know, what all we are going to see today in the slides is structure of human cells. As you all are familiar with it, it is nothing new. Okay. Then composition of the cell membrane, function of the membrane, and the fluid mosaic model, which is a important topic for this. This is as far as concerned exam point of view. This is an important question. You may get a short note on fluid mosaic model. Okay. Now, what is biochemistry? As this is the first class of biochemistry for you all. So, what do you mean by biochemistry? What do you understand? Yes, it is the application of the chemistry of studying the biological processes in the cellular and molecular level. Please come sit down quickly. Now, what are the principles of biochemistry? That is, we study the cell. Every time we study the cell, what do you mean by cell? That it is the basic structural unit of the body. Okay. Now, there are various chemical reactions going on in the cell, and they constitute the basic process of life. Okay. Now, reactions, for example, you must be knowing glycolysis. You know, you are familiar with glycolysis. Okay. So, it is found in almost every organism. Okay. And basically, the molecules which are present in this are the carbon, hydrogen, oxygen, protein, lipid. All these are familiar terms. Okay. DNA, RNA, you all have studied. Okay. So, this is the basic. And what all these chemical processes and reactions lead to? They lead to the growth, reproduction, and development of the organism. And the data which is contained in the cell is in the form of DNA. Okay. Then cell, it is the building block of life. It is the smallest living unit of the organism. Then it is capable of growth, reproduction, use of energy, and respond and adapt to the various environmental conditions. Then cell might be very small that we cannot see with our eyes, and we need a special aid, that is a microscope and all that, and it may be very large also. Okay. Then it may comprise of, there are unicellular organisms as well as there are multicellular organisms. Okay. So all the structures are made up of cells. Now regarding human cell, you know this is the structure. It is there are membrane and membrane bound organelles are present. Cytoplasm is there, built nucleus, nucleolus, everything is there. Okay. You all are familiar with the organelles? Then there may be prokaryotic cells and eukaryotic cells. The main difference being that the prokaryotes, they lack the nucleus and the membrane bound organelles. And in case of eukaryotes, they are having a well defined nucleus, nucleolus, and the organelles are well membrane bound. Okay. Then this is the structure, rough diagrams. You can appreciate these diagrams, that this is the DNA content or the nuclear content. This is the genetic material which is lying this. Can you appreciate this? Yes or no? Okay. So this is just lying in the cell. So this will lead to the so-called multiplication and the continuation of the product to the next generation. And in case of eukaryotes, you can see that there is a well-defined nucleus with proper nuclear material and well-defined organelles which are membrane bound, and they are responsible for particular functions. Okay. Now this is the difference. You all know this is prokaryotes. The examples are bacteria and cyanobacteria, and in case of eukaryotes, they are the plant and animal cells. Then in prokaryotes there is lack of membrane and nucleus, and here it is well defined in case of eukaryotes. Then the genetic material is just found in the cytoplasm. It is not well defined, and here it is proper bound in the proper nucleus and nucleolus. Then in case of They lack a membrane-bound organelles in case of prokaryotes, and in case of eukaryotes, every organelle is membrane-bound. Okay. Then there is presence of ribosomes which will carry out the various processes, but they are simply scattered in the cytoplasm. Whereas in case of eukaryotes, they are properly placed on endoplasmic reticulum. Okay. And in case of cellulose is absent in the cell wall in case of prokaryotes, and in case of eukaryotes, they are well present. Then these cells are simple and smaller in size, and in case of eukaryotes, they may be varying in size as well as complex. Then the various organelles, as you can see, the plasma membrane, it is lipid and protein and carbohydrate complex, and it acts as a barrier. It can provide as a barrier and containing the transport and signaling system. Okay, you all have studied about ion channels and receptors. You all have well read with that. So these membranes are containing. So this sentence means that they act as a barrier. They do not allow everything to pass into the cell, as well as they contain a special arrangement for the transport and the signaling system. Okay. Then nucleus, that is double membrane surrounding. It is encircling the chromosomes in it, and the nucleolus, and the pores allows a specific communication with the cytoplasm. This is also double layered, and there are certain pores. Can you appreciate in this diagram? Okay. Then nucleolus is the site for synthesis of RNA. Then mitochondria, as you all know, what is the function of mitochondria? Energy production. Okay. Then chloroplast or the plastids, again it is responsible for the photosynthesis in plants. Okay. Then rough endoplasmic reticulum. Why it is called rough endoplasmic reticulum? Because yes, ribosomes are present on the surface and they give it a rough appearance. Okay. Then smooth endoplasmic reticulum. They are smooth. They do not contain anything rugged on their outer membrane, and they are concerned with the synthesis and metabolism of lipids. Okay. Then Golgi apparatus, that is, they are arranged in stacks. Okay, and vesicles move between the stacks, and while they are moving, they lead to the processing, that is the post-transcriptional. You all have read about this post-transcriptional modification. Yes. So all these things are taking place over here. Then lysosomes, they are membrane-bound organelles, and they contain substances, yes, enzymes and all, so that they can help in degradation and in digestion and all those things. Okay. Then vacuoles, they are again membrane-bounded bags, and they contain water and storage material in case of plants. Okay. Then peroxisomes and microbodies. Peroxisomes, they help in the production as well as degradation of hydrogen peroxide. There are many reactions taking place in our body which we will be going to read about these things in further classes of metabolism. So these in these chemical reactions there is production of hydrogen peroxide also. So these hydrogen peroxide production as well as degradation both can occur over here. Then cell wall, as you know, it is the additional lining on the outside of the cell membrane in case of certain cells. And cytoplasm, what is cytoplasm? It is the material which is present. This consists of the liquid portion as well as the membrane bound organ. Okay. This is your attendance. Okay. Then cytoskeleton. Cytoskeleton means just like we have a skeletal structure, these cells also have a skeletal structure which gives it the shape and the ability to move around. Okay, and these are made up of microtubules and microfilaments. Then basically what we are going to study are the biomolecules. Biomolecules means they are the small building blocks. Just like cells are the building blocks for tissues, so these biomolecules are the building blocks of the cells, and they constitute the water content of the cell might be ranging from 50 to 95%. Apart from the water, there is presence of ions such as sodium, potassium, calcium, and the organic substances, that is carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. Okay. Then what is the composition of the cell membrane? It is a double layered structure. Okay. It is double layered structure consisting of the lipids and the protein molecules. Okay. Then lipids में क्या होता है? Phospholipid. Phospholipid means to the fatty acid there is attachment of the phosphate, and this leads to the formation of amphipathic molecule. Amphipathic molecule मतलब hydrophilic and hydrophobic both ends are present. Okay. Then in between the cell membrane there is also the presence of polymers. Then proteins, they form the integral membrane proteins and the peripheral membrane proteins. And in case of when they lie on the outer surface, they are peripheral, and when they are located in the interior, then they are the integral membrane. That is, we cannot detach them. And in case of peripheral membrane proteins, the advantage is that they can be detached also. In further metabolisms, you will study about this importance of this ability. Okay. Then transmembrane proteins, they serve as channels. Channels का example, ion channel. Okay. Then carriers, certain substances which cannot pass through the cell membrane, they have to be transported with the help of carriers. In case of lipid metabolism, you will read this, okay, in the upcoming classes. Then pumps, yes, sodium potassium pumps. Then receptors, okay, and LDL receptors and insulin receptors, very common, isn't it? Then functions of the membrane proteins. Now what are the functions? As a transport substance, they act as a example maybe the membrane receptors. Then immunoglobulins, that is the integral proteins of the lymphocytes. Okay, they help the cells to fight against the any organism which is allergen or any pathogen. Okay, so they are the immunoglobulins. Then proteins of mitochondrial membrane, that is, they help in the production of ATP. And in case of RBC, there are special proteins which are known as the encheirin and spectrin. What is their function? They maintain the biconcave shape of the RBC. And glycophorin, they help in recognizing which blood group is there. Okay, so they are the blood group substances. And anion channel proteins, that is, they help in the transport of carbon dioxide via the blood. So, in this way, RBC is very helpful. Okay. Then factors stabilizing the lipid bilayer. What are the factors affecting? So they are the hydrogen bonds, the hydrophobic interactions, and the van der Waals forces. And functions of the lipid membrane, that is, it helps in maintaining the fluidity, the asymmetry, the selective permeability of the membrane. I hope these terms are, you all are familiar with these terms. Nothing new. Okay. Then carbohydrates, extracellular surface may contain some carbohydrate substance, and it is also known as the glycocalyx. Again, this is very helpful in blood group substance. Then fluid mosaic structure, that is the fluid mosaic model. And this, what does it mean? It means that the membrane proteins float in a sea of lipids. Okay, and it was proposed by Singer and Nicholson in 1970. Then fluid mosaic model, this is the structure. These are the phosphate heads which are hydrophilic, and these are the phospholipid tails. These are the tails which are hydrophobic, and these are the transmembrane proteins. These are the transmembrane proteins or the integral proteins. You can appreciate very well over here. These cannot be removed. Okay, and in case of these are the peripheral floating aminolipid compounds, that is the proteins which are present on the outer surface or the inner surface. They can be removed, detached, but these ones which are through and through, they cannot be detached. Then membrane functions, that is, what are the various processes by which transport system acts? That is the passive transport without the energy expenditure. They include the diffusion, that is movement of substance from higher concentration to lower concentration. For example, that would be occur in case of water and gases. In case of facilitated diffusion, that is, small molecules are transported with the help of specific carriers. Example is glucose and amino acid carrier proteins. Then osmosis. Osmosis is movement of solvent through the semi-permeable membrane from region of lower concentration of solute to the higher concentration of solute. Then active transport. Active means against the chemical or the electrical gradient. So definitely it will need the use of energy. Then primary active, secondary active, carrier transport and vesicular transport. So primary active में sodium potassium pump, calcium pump, potassium hydrogen pump. Okay. And in case of secondary active transport, what do you mean by secondary active transport? Yes. That is, energy is being used, but not at the point of utilization. Means wherever, like sodium potassium pump, what is this pump acting for? It pumps sodium out and potassium in. So this energy is being produced at that site only, but with the help of this, along with sodium, glucose is also being transported. So no energy is directly utilized, so we are not using this pump for transport of glucose. But because of the activity of this pump, glucose is also being transported. So energy is indirectly being used. Okay. Then carrier type of transport, that is uniport, symport and antiport. Uniport means one directional. Then symport means yes, two directions it can take place. Two substances can be transported simultaneously, and antiports means one will be yes in opposite directions. Okay. So then vesicular transport, that is the process which the cell undergoes, that is endocytosis, exocytosis, endocytosis, exocytosis. Endocytosis में pinocytosis and phagocytosis. What do you mean by pinocytosis? Cell drinking and phago cell eating. Okay, so phagocytosis, some solid substance would be taken in, and in case of pinocytosis, mostly liquid portion is there. Then receptor mediated cytosis means that they would be taken inside the cell only at a particular place where the receptor for that particular substance is present. And how do we recognize these? By the presence of these special substances or protein particles which are present on the inner side of that. So this is a pit. You can appreciate this pit over here. Okay, pit-like structure. So this is the receptor. Then exocytosis, whatever substance is being produced inside the cell, it is packaged in the vesicle or the bag-like structure, and then this vesicle will merge with the cell membrane and would lead to the exocytosis or the transport of substance from interior of the cell to the exterior. Is that clear? Then whichever what reference books I have used is Satyanarayan textbook and MD Rafi. Okay? Thank you. I hope the lecture